Okay, we're officially logging in now. And then we should see, now we see the participants coming on. All right, so we're just going to give folks a few more minutes to join, and then we'll kick off this webinar.
Oh, I can see. Yeah. I can't see you. Oh, yeah. All right. Hey, Dan is here. Dan Taylor is here. Ooh. Hello, Dan.
It's good to see everyone joining.
Hi, Patricia.
All right. Well, I want to make sure that we give Professor Bonifer and Dr. Kellaway enough time. So I'm going to go ahead and kick this off. Good morning, good afternoon, depending on where you are. Welcome to our RRP hosted webinar series. I'm Katrine Erickson, Executive Director of the RUNX1 Research Program. For those of you who are new to us, we are a nonprofit organization dedicated to supporting patients with RUNX1 familial platelet disorder with predisposition to blood cancers. We do this through funding research, building and supporting our patient community, and facilitating collaboration across the healthcare ecosystem to discover treatment interventions that would prevent blood cancer in these patients. I want to acknowledge our wonderful co-funding partners, Alex's Lemonade Stand Foundation and Leukemia Lymphoma Society, and our newest partners, the Mark Foundation for Cancer Research and Evans MDS. Without them, we would not have been able to fund or commit to over $7 million in research funds over the past 5 years. We've started these research webinars last year during COVID, and we found them to be a great way to keep our RUNX1 research community connected and advancing on the research and the questions moving forward. So we will continue to host these throughout the year. So please look out for additional invitations. In fact, we'll have our next webinar end of April 29th. This will be with NIH investigators, Drs. Pam Guerrero, Keith Sacco, and Kathy Calvo, and they will present their latest research on findings from the ongoing RUNX1 natural history study. And Dr. Guerrero and Sacco will present immunological findings from patients, and Dr. Calvo will present an in-depth analysis of the bone marrow evaluations she's conducted on these patients. So as a quick reminder, the structure of our call will be quite simple. We'll start with the presentation, and then we'll end with a Q&A session at the very end. Please use the Q&A icon when you submit your questions. And refrain from asking questions through the chat function. Please go ahead and use the chat function for other purposes. And with that, I think we're ready to move forward. So I would like to introduce Professor Connie Bonifer, who I think many of you know. She is the chair of experimental hematology in the Institute of Cancer and Genomic Sciences at the University of Birmingham. Her lab works on gene regulation in the hematopoietic system, with a particular focus on how mutant transcription factors like RUNX1 reprogram the chromatin landscape and gene expression of hematopoietic stem cells. Professor Bonifer has dedicated years of her career to studying mutant RUNX1 and its role in AML. And we are very grateful for all of her work and truly excited to hear her present her latest research findings. Alongside her is her very talented postdoc, who is a bioinformatician whiz and also an expert in ES cells. Dr. Sophie Kellaway, and she is the first author on their recent paper entitled Different Mutant RUNX1 Oncoproteins Program Alternate Hematopoietic Differentiation Trajectories. So with that, I'm going to turn it over to you, Professor Bonifer. Thank you for being here.
Thank you very much, Katrine, for the lovely introduction. And I think without much of an ado, I'm going to, I think I should share my screen first. I will start my presentation and let me know if there's a problem. There shouldn't be. Okay, can you see me and hear me properly?
Yes, we can see.
Okay, fantastic. All right, well, Katrine, thank you very much for inviting us to give an overview about the work that the lab has done in the last few years. And also for the kind attention of all the delegates that have joined this call. And my talk will be about how variant transcription factors apparently program the epigenome. And I don't have to explain this to all of you, that transcription factors are really the most important molecules when it comes to driving cell fate transitions from one cell type to another. And this hematopoietic hierarchy picture, which I don't have to explain in more detail, just gives you an overview about the most important transcription factors that drive the development of stem cells, such RUNX1 and SCL-TAL, but also transcription factors which are important for further downstream developmental pathways, such as GATA1 and CVP-alpha. Now, what is important to know regarding transcription factors is the fact that they can form gene regulatory networks. And the reason why they can do this is because they can bind to each other's genes and regulate their mutual expression. And what you see here on this scheme is that is a subnut network for myelopoiesis, which consists of myeloid development from stem cells, which consists of a recursive network of GATA2, FLY1, and SCL, which then again regulates the master regulator RUNX1, which then in turn regulates the further downstream genes such as CVP-alpha, beta, and PU1, and so on. But what is important here to realize is that these networks, they are very highly dynamic. And there's various points in development where signals can come in that can kick off changes in these networks. That can be signaling responsive transcription factors responding to growth factor receptor signaling, cell-cell contact, wind, notch, and so on. And eventually also there will be also feedback regulation from genes that will make sure that development doesn't overshoot. Now, this is a very nice, and I stole this particular scheme from Eric Davison, who is an absolute pioneer in thinking about decision hierarchies and transcription factor networks. And in principle, all development can be described like in a decision hierarchy where you have the initial inputs, where a gene regulatory network is specified that tells the cell which differentiation pathways the cell doesn't want to be in. And then these kick off the further development, which then is regulated by gene regulatory networks that define which cell type within this pathway these cells want to be in, which are the differentiation drivers. And at the end of this, you have the actual organism and the specified cells, which then that have specific gene regulatory networks that define which kind of cells they are. And in principle, and the question is, how do you determine the structure of these networks? And there's two ways to going about it. One is the classical way how Eric Davison used to do this. They used to perturb the expression of a putative regulator and then look at RNA-seq gene expression changes, which then occur in different patterns, from which then it can be inferred which the regulatory hierarchies can be inferred. But nowadays, we have the ability to actually look at actual binding events by either chip or by in vivo footprinting for factor families, which then can be annotated to their rightful promoters by high-C experiments. And using such technology, we can then see how one gene regulatory network as a result of signaling input then turns into another and specifies a different type of cell. So why is this important? Well, it's important because cell differentiation can go wrong. And this is what happens in leukemia and in other cancers. You start out with a stem cell, which then suffers from the first hit and creates a pre-leukemic situation, and then turns into AML blast cells, which then further evolves into by clonal selection or relapse and acquiring different mutations. So the mutations that we find in AML are usually those of transcription regulation in some way or another. So this is transcription factors. These are epigenetic regulators which cooperate with transcription factors or set the stage for a certain chromatin structure, but also mutations in signaling molecules that then also send out chronic signaling that terminates at signaling responsive transcription factors and chromatin component. So when all cancers in general are therefore is a disease of epigenetic reprogramming and transcriptional malfunction. So in the last 10 years, we have used multiple multi-omics analysis using patient samples to try and work out how the different mutations, which are at the basis of the changes that we see in AML, how they impact on cell differentiation and also on transcription regulation. So we collected a large number of AML samples from patients with defined mutations. We measured mRNA chromatin mutations, promoter cis-regulatory elements, and performed the integrative data analysis. This is all published, so I'm going through this quickly. We looked at at a limited set of transcription factor mutations, most of which come from the core binding factor complex, so that the RUNX mutations themselves, the TA21 inversion 16. We also looked at CBP alpha double mutations and FLT3-ITD NPM1 mutation. And when we then cluster all the different cis-regulatory elements, which we map by DNase I hypersensitive site mapping, we realized, and we did the same for gene expression, it was very obvious that both features cluster exquisitely according to the patient class. So the FLT3-ITD is clustered together, and the CBP alpha TA21 and the inversion 16 clustered together as well. And they also formed subgroups, whereby it was obvious how that some of the core binding factor mutations were very close to what the CBP alpha mutations. The RUNX mutations being somewhat heterogeneous, clustered in as a separate group. Now, what can you do with this analysis? Well, you can identify AML subtype specific cis-regulatory elements, which exist as DNase I hypersensitive sites. So we identified some which are common to all AML. We identified some which are specific for the TA21. We identified some which are specific for core binding factor AML and CBP alpha mutations, FLT3-ITD AML, and also some that were exquisitely clustering in the group together with the CBP alpha mutation. And so the question is then, which transcription factors are then involved in regulating the activity of these AML subtype specific elements? For that, we turn to something that my group has been doing for many, many years, first in a gene specific way, but then also now in genome wide, by doing digital footprinting, which essentially highlights regions in chromatin which cannot be cut by DNase I because transcription factors are occupying these sequences. And we developed an algorithm called Wellington, hence them boots, and which shows that some specific sequences are exquisitely protected from digestion. And for example, this is an AP1 motif, which is protected from digestion in FLT3-ITD past blasts, which is this black region here. So we know that these factors are active and they're actually doing something. Now we can then, of course, do this whole in genome wide and for all of the patients. And what we then can identify is specific motifs which are occupied in specific types of AML. Now, in this case, this is the TA21, and you can see that, for example, here, these are all the factors motifs which are occupied in this particular, in the elements which are specific for that type of AML. And then we can see it's ETS, TAL, and also PAU4F1, which is a factor that's normally not expressed in these AML at all. If you do the same for a FLT3-ITD, if you do the same for the FLT3-ITD patients, which are these elements here, you get a completely different signature. So we get HOX motifs occupied, FOX motifs are occupied, RUNX very strongly, PBX, and a lot of AP1 motifs, and also here early growth factor response factors, family factors. So what we can see from all of this analysis is that AML subtype specific cis-regulatory elements are regulated by different transcription factor families. So the question is, which genes do this cis-regulatory element belong to? And for that, we turned to promoter capture high C, which we now did in 4 different types of patients in the FLT3-ITD, the TA21, and also the CBP alpha double mutation. And the first thing, and as you can see, the overall structure, like the tab structure or something, is more or less the same between all of the patient samples. However, what we noticed very quickly is that in order to analyze the data properly, we can turn to our DNase1 hypersensitive site pattern that we identified. And we saw that pretty much the majority of all hypersensitive site in one way or another participate in interactions. And so we could throw away all the other interactions which are mediated by other factors such as polycom and concentrate on the hypersensitive sites. And this is an image from the FLT3, from the CBP alpha locus in the FLT3-ITD and normal cells. And you can see that in normal cells, we do have certain interaction here, and we have additional ones in the FLT3-ITD with elements that are popping up upstream of the CBP alpha gene, which appear as hypersensitive sites. So when we merge all the DNase1 hypersensitive sites that we find in these 4 different samples, we notice that the vast majority is shared, because after all, we're looking at a similar cell type. And also, when we then merge all the hypersensitive sites from all of the patient samples that we've ever analyzed, we noticed that this high C data set covers about 95% of all interactions that we see. So what that means is we can link footprints and cis elements for the vast majority of genes to their rightful promoter. And now we can construct gene regulatory networks. And this is one of them. This is the TA21 specific network with the factors families linked up to each other. And this is just the transcription factors linking to each other. So these are the factor families which are in the circle binding to the same motif. And if there's an arrow to a specific gene, that means that one factor, any of these factor families binds to this particular gene. So, and you can see in the TA21, for example, the HOX genes are not active. And you can see which genes are active just by the color of the expression, which when red is highly expressed. So you can have an educated guess which factors are actually important. And what's also interesting here is, and this is the FLT3-ITD, here the links are completely different. Here the HOX genes are active, but and show links, but we can also see links from other factor families, and especially here from the AP1 family to a number of others. So what that means in conceptually, and of course, this entire network regulates the non-transcription factor gene with the AML specific footprints. Okay, so all the downstream genes are one way or another are linked to this network. And I'll show you about one of these modules later. Now, what that means is that myeloid differentiation in AML is not just blocked, it goes actually sideways. And the cells adopt a new identity that's shaped by selection. And we can, and in new data or new analysis that we've done, we now followed this during subclonal evolution. And the rules we discover are really seriously interesting. So, and the central hypothesis of our work is that we think that uncovering these networks or these will highlight those AML subtype specific pathways which are required for tumor survival. And I'll show you two examples for it. One example is the AP1 transcription factor family, which forms an important node in all AML subtypes we've analyzed so far. And for two of them, we expressed a dominant negative FOS, which blocks all AP1 activity in cells, and transplanted these cells in mice. And we could strongly, and this was an inducible construct, we could strongly block AML tumorigenicity in this system. The other one is your good old friend RUNX, which is also part of this network. And you can see there's a great many arrows coming in from other transcription factors into the RUNX, into RUNX1 here. And RUNX itself is the motives for RUNX are in pretty much are enriched in the majority of FLT3 ITD specific cis regulatory elements. About 50% of these elements, which are specific for the FLT3 ITD core, are bound by RUNX. And this, what we call a trumpet plot, shows you the actual number of genes which are downstream of RUNX, which are all bound by RUNX. And we have both the footprinting and the chip data to show this. Now, what we still have a very nice collaboration with John Bushwiller, and who's developed the small molecule inhibitors for the RUNX binding, and which disrupt CBF-beta RUNX interaction. And when we put those small molecules on FLT3 ITD cells, what we see is very nicely is that in the viability of these cells strongly decreases if we add more and more of the inhibitor. If we do this with normal cells, we only find an increase with the highest concentration. Now, and this is a really cool experiment because this is all patient cells, and this patient has a mutant in both RUNX alleles. So RUNX is not functional. But although it does have a FLT3 ITD, it also has this double mutation. And these cells really are not responsive to, or not really responsive to the RUNX inhibitor at all. So this tells you very nicely that interpreting this gene regulatory network can really help you to try and identify transcription factors absolutely important for the survival of these networks, but also will also identify downstream genes, some of which may code for products that are druggable. Now, the second part of my talk will now deal with the more molecular side of things and how RUNX actually operates. So the question's really, how did the cells actually get there? So it's very clear that different mutations set cells on a different path to malignancy. So the question is then, and we were very interested in this, what is the effect of the first hit? You know, because in patients, we can only see these cells that have already accumulated a large number of mutations. So what's the effect of the very first hit? So how does the very first hit interfere with normal development? You know, what do these aberrant molecules actually do in terms of the molecular mechanism, how they operate? And one way of identifying or answering these questions is the differentiation of embryonic stem cells, and we can now do this both with mouse and with human. So we can, so just to recapitulate for everybody, is you can differentiate these cells into embryo body, you can sort them, put them in culture, and then something developed that's called a hemogenic endothelium, which then undergoes a endothelial hematopoietic transition, where then the progenitors sort of pop out and go into solution. And you can then differentiate those even into the mature cells. This is now fairly mature technology, and a lot of people are working on this, in particular with the human system. Now, one crucial information is that this transition from the endothelial cells to the blood cells is absolutely dependent on RUNX1. And we were interested to try and work out what RUNX actually does. And I'll summarize this very quickly because this is now really published a long time ago. What we found, what we used was an ES cell system that was generated by Georges Laco, and who had expressed the RUNX transgene in an inducible fashion in the background of a RUNX negative ES cells. And when you try and differentiate those cells, these cells are then blocked at this stage here. And when you induce RUNX1, out pop the progenitors. Okay. So we could show that hematopoietic genes are already primed in the hemogenic endothelium by the binding of other transcription factor. And once RUNX comes in, it initiates the reorganization of transcription factor assemblies at key lineage determining genes. And it operates together with other transcription factors and pulls them somewhere else. And this is what you can see in this picture here. What we've done here is we performed chip experiment with and without RUNX induction. And what we observed to our surprise was that these factors then start to move into different directions. And what we then also noticed is when we look at, let's say in this case, and this is a later paper that we published a few years ago, 2018, when we look at FLI1 binding, with or without doxycycline, and we normalize this to the RUNX binding site, which are here, what you can see is FLI is faithfully moving where RUNX is. And we could also show that RUNX binding and FLI1 binding come closer together. So RUNX pulls these factors in. And what does it do then? It then upregulates the expression of hematopoietic genes. And this is just an example for such an analysis where we look at polymerase II occupancy over a idealized gene from the transcription start site to the 3' end. And we could see that without doxycycline, polymerase II is not much occupancy, but then Pol2 comes on. And this is a wonderful marker for actually following transcription elongation, which is H3K2979 a dimethyl profile. You can see that the polymerase marches on, recruits DOT1 and puts this mark on the chromatin. And that is absolutely dependent on RUNX1. And this is the gene expression. And what's also interesting is that a third of these genes are already bound by SEL and FLI. And these are the genes that are essentially activated. And vice versa, when genes are repressed by RUNX1, we're losing Pol2, we're losing elongation, expression goes down, but also these down-regulated genes have RUNX1 bound. So how does RUNX switch on the expression of his target genes? And I'm just giving you a schematic overview because all of this work is published. We have what we find in two groups of genes which are upregulated by RUNX, and some of them have Pol2 already bound and some of them don't. What we find is that we have a binding of the bromodomain protein BRD4 at sites where TAL1 and FLI1 is binding. These sites are acetylated as well. Then once RUNX1 comes in, it recruits FLI1 to its new site, and BRG comes with it. Then mediator is brought in, and we also find a binding of PTEF-B and CDK9, and that is absolutely dependent again on RUNX1. We know that RUNX1 doesn't interact with BRD4. We know it interacts with these guys here, meaning that RUNX drives the endothelial hematopoietic transition, chromatin opening, transcription factor relocation, and also RUNX1 dependent gene expression. And we find such movement of RUNX not just in this particular setting, but we find it in other settings as well. So what we also realized that RUNX1 and BRD4 have a very intimate relationship, although they don't interact with each other. Because if we put these kind of movements and interactions and assemblies of RUNX-dependent transcription factor complexes, that's crucially dependent on BRD4, because if we put a BET inhibitor in here, we block the embryonic, the EHT completely, and we also block the development of progenitor cells. And that's just shown you in the same assay. The POL2 goes down, and we find that when we add a GQ1, we find that elongation goes down, and interestingly, also RUNX binding goes down, which means that these complexes are actually dependent on each other. Okay, so, and this is a schematic. So if we then, in the presence of JK1, we do not get TDK9 recruitment, we do not get RUNX1 binding, and everything stays as it is, and we cannot and RUNX1 actions completely blocked. Okay, so the question is then, in this system, how do the mutant RUNX proteins interfere with this process? And this is how Sophie's work now comes in. And we looked at different types of RUNX mutations, and which are responsible for primary AML, therapy-related AML, myelodysplastic syndromes, and FPD. And the question then is, what are the molecular mechanisms which underlie these different malignancies? So what do these molecules actually do in this system? Okay, so we looked at the R201Q, which in Nancy Speck's paper is called the R177Q. And so we were guided by our choice for these mutations from Martini et al., where Nancy Speck very nicely dissected in the mouse what these different mutations do. So we looked at this one. We looked at one that, so this one can't bind to DNA. We looked at one that has just the DNA binding domain. And this one here is FP, causes a familial platelet disorder. But we also looked at RUNX1-ETO, and we looked at RUNX1-EVI. So essentially, we compared 4 different mutant RUNX proteins, which all can bind to RUNX binding sites, and then asked the question and created embryonic stem cells with those, and then asked the question, you know, and oops, something happened here. Okay, so we created you have been signed out. Am I still there?
You are,
Katrine,
Connie.
am I still there?
You're here still, Connie.
Oh yeah, okay, because my computer just told me that I'm signed out for some reason.
You're still with us.
Okay, good to hear that. Okay, so we created embryonic stem cell expression and expressing all these different mutant proteins, and asked the question, what do we actually see here? Do we see a dominant negative effect, or do we see a perturbation? The first one is dependent on RUNX, and the second one is dependent on downstream pathways. So, and also, most importantly, we asked the question, do all these mutants interfere with the same set of genes? Because after all, they all bind, they all have the RUNX domain. They all should bind to the similar genomic binding sites. And so this is just the biology, all of this. I have to explain what we're seeing here. Essentially, what you need to understand is that differentiation is not entirely synchronous. So we're actually hitting the cells when we induce these mutants at different time points in different developmental stages. But we can still say we can still see what they're doing. So we have basically 3 different types of progenitors in here. We have HE1, which is the hemogenic endothelium, which expresses, which doesn't express RUNX1. This one and expresses TIE2. This one starts to express CD41, which is a RUNX target. And here are the actual progenitor cells, which express RUNX and have downregulated the endothelial markers. So when we look at how how many cells are formed, what we can see is that RUNX1-ETO and RUNX1-EVI both block the hemogenic endothelium, the endothelial hematopoietic transition. And we can see this in a lack of, in a reduction of progenitor cells here, and a block at this particular stage here. The R201Q and the R204X don't really do very much. So the major disruptors are really these two fusion proteins. That's why you never find them, that they're incompatible with embryonic development. However, these others, of course, run in families, and that tells you why. The endothelial hematopoietic transition is not affected, at least at this stage at all. We can form progenitors quite nicely. So, and that just summarizes what we see. However, what we noticed is that the R201Q blocks or has a problem with megakaryocyte development. So we're getting a significant reduction in megakaryocyte development when we induce this particular mutant, which is not so easy to see, but Sophie found a way of how to make this visible. What about gene expression changes? Again, another interesting result. What we can see here is that the R201Q basically doesn't change gene expression at all, or very little. I mean, in these volcanoplasts, in the progenitors, you see a little bit of a drift, but that's all. The R204X is a little bit, has a bit of an effect in the hemogenic endothelium, and upregulates a few more genes and downregulates very few. Whereas the RUNX1-ET and RUNX1-EV have a massive impact on gene expression changes. And when you then look at what these genes are, they're all different. There's barely an overlap between the gene expression profile changes between all of these different mutants. The molecular mechanism behind this must have something to do with the particular architecture at these sites, which I pointed out to you in a more generic fashion, at these particular sites when the mutant hits, okay? And it's quite mind-boggling if you think about how complicated the regulation is at these particular sites. All of these mutants change gene expression in their own way. Now, what do they actually do with respect to RUNX? Well, if you look at and then we perform chip experiments and compared this also to gene expression. So if we look at RUNX1-ETO and RUNX1-EV, we can do chip experiments. These are the binding sites for RUNX1-ETO. These are the binding sites for RUNX1-EV. And here we have RUNX binding before and after induction of the oncogene. And you can see that the expression of these oncogenes has a profound effect on the binding of RUNX itself, and also on gene expression here. And these are just the chromatin sites change as well. Now, for the others, we did not see any binding at all. So these proteins do not leave and cannot be chipped. However, with the RUNX one, with this one here, we saw an absolute dramatic drop in RUNX1 binding once we induced it. And here we basically saw nothing, okay? Which also tells us something how nicely reproducible our assays are. Sophie is really good at that. So here we could really see hardly any RUNX binding. So what's going on here? So what Sophie did then was she performed a proximity ligation assay, which is sort of like simulated here, which means when you have two fluorochromes and when they come close together, they make a light. And the question was then, do these mutant proteins also interact with CBF beta? Because that's what we were looking at. Or does this affect the binding of the endogenous RUNX as well? Okay. So when we go through the different assays, what RUNX1-ETO does is it shows the PLA shows very little changes whether RUNX in RUNX CBF beta interaction of the wild type protein. the good news here is we have an HA tag, so we can actually also see what the transgene does. So there wasn't not many differences. With the RUNX1 EV, what we saw was that and in this case, we're using an antibody against EV. There's again, not much changes. And if at all, we see an increase in CBF-beta RUNT domain interaction here. The R204X doesn't affect the interaction very much, but the dramatic effect was with the R201Q. Here, what we have is a drastic downregulation of CBF-beta and RUNX interaction. And when we look at the interaction between the transgenic protein, expressed protein, or the ectopic protein and CBF-beta, we saw very little. And this slide just summarizes the results. Here it goes down, here it stays the same, here it's more or less the same, and here it goes up, okay? So obviously there is a massive effect of the transgene on RUNX binding. So the question is then the FPD mutant only, and this is in the total absence of changes in gene expression. We only have 2 genes that actually change gene expression. So how can we explain this drastic phenotype, right? So maybe it has something to do with the opening up of chromatin. So what we've done here is we compared the different hypersensitive sites that we mapped as well in the different induced and non-induced conditions between the R2OX and the RUNX1 And what we realized is that each of these transgenes changes the chromatin accessibility in a different way. And that is also true for the others. So maybe what happens is that we are blocking a differentiation pathway. And what really happened here, and this has been shown, we can show this directly. So what we've done is we are looking at sites, at hypersensitive sites, minus and plus docs, which were forming or were being lost after induction. And then we asked, and then we had a lovely data set which just came out from Ross Hardison's group and others, which identified specific sites for monocytes, erythroblasts, megakaryocytes, B cells, and so on. So we asked the question, with all the different with all the different mutants, will these hypersensitive sites, which are specific for these cell types, are they being formed? And this analysis, which shows you that there is a problem. So although the gene expression doesn't change, what does change is the reorganization of chromatin, which primes the cells to make a mature cell type. And this is a bit like a GSEA here. Here we have the lost sites, and here we have the gain sites, and when we're inducing the R201Q megakaryocyte sites are underrepresented. The same is true with the RUNX1 as well. But what goes up is the erythroid-specific sites. And we can summarize this for the different mutants individually. We also did some histone modification chips to try and work out what's happening with histone acetylation. If we induce RUNX1 we find that it interferes with RUNX binding. We lower histone acetylation at the sites. And we also have an increase in monocyte and myeloid progenitor development. And curiously, also we see more B cell sites. The RUNX1 completely messes up the entire differentiation pathway because the EV protein itself also programs a different set of genes. So we're losing histone acetylation, but we're also gaining at different sites. And it also binds with CBP-beta and sequesters some of that. And we're getting an increase in megakaryocytes, an increase in B cell. TMP stay the same, and monocyte and erythroid go down. The R204X doesn't do very much at all. everything's fine, but we're losing histone acetylation at these sites. And that may be causing a long-term effect on how genes are being activated, and that may contribute to AML development. The R201Q has a very clear downregulation of megakaryocyte sites. Everything else stays the same, but erythrocytes go up, and we can see this directly. And when we do a GATA1 chip, we find that these cells have an increase in GATA1 binding, which explains why we have more erythroid sites than megakaryocytic specific sites. Okay, so I'm coming to close here now. And just in conclusion, these two translocations both alter the binding of endogenous rungs very rapidly. But RUNX1-EV also induces de novo sites and acts as a dominant negative to EV1. And Sophie has published this in an article in Hematologica separately, and where this phenotype is analyzed in more detail. The R201Q causes a global reduction of RUNX1 binding, which is absolutely dramatic, which is associated with reduced CBF-beta interaction. And that leads to increased GATA1 activity and a shift towards the erythroid branch. So somehow the cells make it through, but RUNX is massively impaired. And this imbalance is likely to be there from the very beginning. I'd be so curious to see if how this manifests itself in the cells of the patients. Now, the R204X does not act as a dominant negative in terms of CBF-beta interaction, but we're losing histone acetylation and in some sites, and RUNX, we find there's still RUNX binding, but it still causes myeloid leukemia. So it is probably a mixture of haploinsufficiency and this impact on the chromatin structure. So, I don't want to stop without making sure that the people who've done the work are acknowledged. Much of the work in the earlier stages was done by Salam Asi and another postdoc, Rosario, and Dan Coleman, who's right here. And the RUNX story in the middle was done by Jane Gilmore. She's unfortunately left. And Sophie has done all the ESL work, and Salam and Peter Keen have done all the data analysis, and Sophie did a lot of data analysis herself as well. And I will stop now and ask questions. And I made it in 2 minutes. I'm surprised. Brilliant. Okay, I'm stopping sharing now.
Thank you so much, Connie.
Sophie and Sophie, can you put your picture on?
Thank you, Connie. Thank you very much. Beautiful work. Well done, Sophie. We've got lots of questions, so I'm going to start to read them off. So I'll begin with the first here. The R201Q mutation gave you an erythroid megakaryocyte bias. What is the evidence for this aside from GATA1 association? GATA1 is critical for megakaryocytes as well. And this question was coming from Diane Kraus.
Sophie, do you want to answer this, or do you want me to answer this?
I can answer. So we can see it in both the colony assays and in using our ATAC sites to look for the actual enrichment of specific cell type specific sites. So it's sort of like a gene set enrichment analysis. And we could see an enrichment of the erythroid sites at the expense of the megakaryocytes in that. But we could also see it in the actual mixed colony assays.
So we have an increased GATA1 binding. Doesn't mean that GATA is not important for megakaryocytes, but I think RUNX does more than just regulating GATA1. Yeah? I think it regulates other factors as well. And I mean, it's well known that you need high levels of RUNX for megakaryocyte development. Yeah, not so much for erythroid development. I think it's the balance that's just disturbed. Okay.
The next question is from Irene Papapetro. Hi, Connie, very interesting work. 3 questions. Oh, wow, 3 questions. Okay. 1, how were the ES models made? How were RUNX1 mutations and fusion genes introduced to the cells, ectopically or endogenous loci? And then I'll let you answer that, and then we can go to the next.
Okay, Sophie, can you please answer?
I will jump in again, yeah. So the models that we use, they're doxycycline inducible, and they're in the, I've forgotten the name of it. Sorry, my mind has gone completely blank. They're in a stable locus anyway. This is a cell line that was published.
It's the Michael-Kyber system, essentially.
Michael-Kyber system.
Yeah, so it's a landing site where you can put in the where you can place various transgenes in a doxycycline inducible fashion. So it's not an endogenous gene, but we can control the expression levels very carefully. Works really well.
Yeah.
And it's also one of these sites where you can where it's really expressed in every cell. So we don't have genomic position effects or anything. Yeah, so it's not an endogenous locus. And that comes probably your next thought is, yes, the wild type load, we titrated, or Sophie titrated the expression level to pretty much the same as the endogenous rungs.
Yeah, so rather than it being a heterozygous knockout or anything like that, site mutation, we just actually induced it on top of it.
Yeah, so we could also see the interaction between the two the proteins, because if you change the levels, you can see quite an interesting effect here of the interaction between those proteins, which we haven't talked about.
What the follow-up question is, what cells exactly during the differentiation were used for the analyses, the gene expression, the chip, etc., and were they selected with some marker? Were they hemogenic endothelium, and how heterogeneous?
So they were the hematopoietic progenitors. They're 95% or more c-KIT positive, CD41 positive. There's going to be some heterogeneity just because of the stage they're at, but we did sort it to get as pure a population as we could.
So you remember I showed you the profile with the HE1 that goes from HE1 to HE2 to the progenitors. So we sorted these individual cell populations, right, based on Tie2, KIT, CD41.
Yeah.
So anything endothelial is Tie2 negative. Anything hemogenic endothelium is Tie2 positive, CD41 positive, and anything progenitor is CD41 positive, c-KIT positive.
So the final question is, how relevant do you think these findings are when you're thinking about the role of RUNX1 in the hemogenic endothelium and extrapolating that to RUNX1 and its involvement in AML?
Well, I think the biochemistry doesn't lie, So the biochemistry is what it is. If we can see and I don't think this will be any different in whether we're looking for mouse or whether we're looking at human or whether we're looking at anything else. I think what was very telling was really the fact that these proteins really behave all very differently, and that absolutely mirrors the clinical phenotypes, which was staggeringly interesting. Even in this system, you could see what cell type is affected. And it's very much like, and I mean, we've been working on RUNX1 for the last many years. And it's amazing how much the biochemistry reflects the clinical phenotype here. But in its purity, it's just the fusion protein we're looking at and not anything else. Yeah? So the fact that the R201Q completely interferes with RUNX binding, or with much of RUNX binding, for us, this was a real surprise, actually. Mm-hmm. And I'm sure that in the patients, there has to be some compensatory action, sort of mitigate this in some way or another, But it makes me cranky,
It's a little, I have to admit, it's a little frustrating because there are so many different mutations. When we look across RUNX1 FPD, the patient population, I think there's been over 100 different mutations described across the RUNX1 gene. And so that makes this very complex when you think about the fact that some of these mutant proteins could be behaving very differently.
Yeah, I think maybe somebody should just do a screen of something, just put them all in and see what happens. Because I mean, we really had to go through great lengths in order to see this. You cannot do this in a cell line, you know.
Yeah.
And the reason why that is, whenever you have an inducible cell line where you ectopically induce an oncogene, that interferes with its gene regulatory network, which is fixed because it's a cell line. And if you then put the oncogene on, they either go into apoptosis or they go funny. And it's completely irrelevant what you see. But you have to actually do it in a context where normal differentiation takes place, right? And then you can send the cells into a different direction.
Yeah. So we have another question from John Bushweller. For the RUNX,
hi, John.
For the RUNX1 mutant that loses interaction with core binding factor beta, presumably the cytoplasmic level of CBFB goes up. This could alter filament function. Any sense that it be part of the phenotype?
Sophie, say something. I think there's some cytoplasmic state. Is it?
I mean, I checked the immunocytochemistry for CBF beta. It is in the cytoplasm as well as the nucleus, but I didn't try to quantify it. So maybe.
The next question is from Patricia Ernst. Do you think that the translocations occur in the hemogenic endothelium during human development? And if so, any idea what can be done to, let's see, how could you block the escape or change the transition from hemogenic endothelium, I think is what she was.
Well, if you have RUNX1E2 or RUNX1EV, you won't get progenitor cells. So this is incompatible with hematopoietic development. This has been shown a long time ago in the mouse, very clearly. You know, once you express these things in prior to a hematopoietic to stem cell development, you don't get stem cell. It's a complete block. When you express it afterwards, it's different. Right, and this is the devious thing about these familial mutations, is that they seem to be they allow stem cell development to occur, but then it becomes derailed. Yeah. I guess that was the question.
I believe so. Yes.
Yeah, I mean, if you express RUNX1E2 later on in progenitors, right, or in RUNX1EVO, then you get your normal, hyperplasia and stuff like that. The cells get more increased. They have a cell cycle problem, and you get increased replating activity and survival and all of that. We've studied this extensively. There are several publications from my lab that show that.
There was a question from Mary Ellen around the timing of when the RNA sequencing analyses were conducted, and then additionally, how many of the RNA-seq changes do you think are directly due to RUNX1 changes versus indirect due to other changes?
We don't know, and I mean, there's this lovely paper from Scott Hebert with the degraders, who shows that if you have a rapid response, I mean, Sophie, how long did you take? It was overnight, right?
Yeah, it was 18 hours, which 18 is basically
induction.
We could get it.
Yeah.
Once the
So
levels...
we don't know. I mean, we've always been very open about the fact that when you manipulate these levels of oncogenes, and that you will get a lot of direct and indirect changes. Yeah, whereby not all rapid changes are actually, let's say, tell you anything about the network as such, because you may have to, not all, if you take an oncogene away or you add it, you need some time to assemble the whole machinery, or disassemble. Right. So I don't think this is a black and white, but no, I mean, it will probably be direct and indirect, both of it. And it's very difficult to do it unless you really work on the single cell level. And if I had more funding, I would do that, and to see what the kinetics actually is of genes coming on and going at the single cell levels after induction for a time course. If somebody gives me a $20,000, I'll do that. please.
What I can just add here is only about 30% of the genes which change in total were RUNX1 chip targets.
Yeah, that we can say that. Yeah, true.
Right. Okay.
So there's definitely both.
Yeah, you've got both processes happening. So I'm going to close with one final question in the last minute.
Oh.
I noticed that we have patients on the line. So this is, I'm going to ask you to think about how does your work, what you've discovered, actually impact and affect the future therapeutic initiatives for RUNX1 FPD patients?
Well, I have wondered about this, and I always, I mean, in principle, what's important here is to try and make sure that it is understood that if you want to do therapeutic intervention, you really have to understand what precisely is happening in the cell. And I think trying to understand how these mutant RUNX proteins really set things in motion, in my opinion, is extremely important. And I mean, what I would like to do is really to look in patient cells to see whether some of those reductions, let's say in CBF beta interaction, that we see that there as well. You know, it appears that one of the apparent interaction is being degraded. Question is, and I know people have tried that, whether we could, if we stop that degradation happening, whether we could rescue the phenotype, you know. And but then, of course, you know, this is do BET inhibitors in some way help, or you know, I mean, there's more mechanistic analysis to be done. Right. And what I'd really like to do is to see whether we can get our hands on patient samples to try and see, you know, how is differentiation affected, what kind of drugs can we put on there, how do the different proteins interact, and can we come up with a unique way of how these mutant proteins interfere with the wild type RUNX? Can we come up with an idea what exactly is happening? Could we rescue the phenotype for the I mean, I'm just speculating here. Rescue the phenotype of the DNA of the R204X mutant by adding histone, by adding butyrate, histone as deacetylase inhibitors, right, for example. I don't know. I mean, this is sort of lines of research that one could think about based on the experiments that we've done.
Great. Well, thank you. I know that what you presented today was years and years of work, and it was really just a beautiful story arc of all that you've learned around RUNX1 and these gene regulatory networks, and the importance of really dissecting how these gene regulatory networks are affected by different mutant oncoproteins, even within one RUNX with one transcription factor. So I really appreciate you taking the time, Connie and Sophie.
My pleasure. Our pleasure.
A fantastic presentation, and we hope to see you all back for our next webinar at the end of April. So please,
please send us an invite.
We will, yes.
Okay.
Look out for the invite, the flyer. Take care, everyone. Bye.
Take care, and thank you for listening. Yeah.